Takfarinas Medani. He's going to talk about brain signal modeling. And while you're sharing your screen, I'd like to also thank you because he's now sitting in California. He had to wake up very early. So thank you, Takfarinas, for that. I will read his biography. So he's a research associate at the Signal and Image Processing Institute in the University of Southern California in Los Angeles. So he received his PhD in electrical and computer engineering and master in science in electronics from the Pierre and Marie Curie University in 2011 and 2016, respectively. So prior to his appointment at USC, he used to work in the Brain Plasticity Laboratory within the Brain-Computer Interface team at the École Supérieure de Physique et Chimie de la Ville de Paris in Paris, as an assistant professor and research engineer with the Alzheimer's Diseases Institute and La Salpêtrière University Hospital in Paris. So his research interests are in the fields of neuroscience, from modeling and analyzing brain activities, including invasive and non-invasive modalities, to their utilization in real cases such as brain source localization, brain connectivity, brain-computer interface, and neurofeedback. He's also contributing to educational research tools such as the Brainstorm software at the Neuro Library. So Takfarinas, thank you very much. I think what we can see now is the presenter view of your screen. I wonder whether you can change the screen so that we can see the full one.
Sorry for that, yeah. How about now?
Yeah, now it's good. Thank you very much.
Okay. Thank you very much, Nicola, for the introduction and for sharing this session. And also thanks a lot for Shauki to invite me to present the work that I'm doing at USC right now. So there is some similarity of my presentation with the previous presentation given by Professor Weir and Francesca, but it's in another frequency range. So today I will present the brain signals, so the origin of the brain signals. So it's just general presentation about the theory, modeling, and the tools, the ones that you cited in the introduction. So yes, as you said, I'm at USC in California, and we are working closely with different organizations in the United States, McGill in Canada, and also with German group, which is at Münster, which is the Institute of Biomagnetism and Biosignal Analysis. So this is just the two logo of the two software that I will show quickly at the end of this presentation. So during this presentation, I will try to follow this line or this outline. So I will show quickly the brain signal recording, and then I will talk quickly about the origin of the brain signals, and also how to model the brain signals. So I will focus mainly on the forward problem, its formulation, and the effect of the geometry and the conductivity, and of course, its resolution. And I will show all the work that I did, part of the work that I did, which is the implementation of all this pipeline in the Brainstorm software, which is neuroscience software, and also all the plugin that we add. And I will end up by conclusion, and of course, I will be happy to answer all of your questions. So first of all, so brain signal recording. So we are in the low frequency range in contrast to what it is shown before. So here we have a patient, which is one Polé, which is Carsten Walters. So here we have two devices. So there is this hamlet here, which contain electrodes that record the voltage on the scale. And there is also this big machine, which is the MEG. So this one is EEG. This is the MEG. And here there is a supra-squid, which are supraconductor sensor that catch the magnetic field that's generated by the head. So as you see here, just a quick video. So each channel is named by a specific name that can help us to localize it on the head or on the sensor, spatial localization. And it depends on the application, but for example, here he's tapping his finger, left finger, and then we record the signals, the time series. And from this, we can display the topography on the sensors as well as on the scalp. As you can see here, we can see a dipolar pattern, which is the moving dipole in the left hemisphere. So he's moving his right hand because we are looking at the left. So from this data, we can, for example, localize and look for what is going in the brain or on the cortex of the brain, and we can display it in different modalities. So this two devices is not just for displaying, so there is a lot of application, and most of them just trying to understand the brain's function. So up to now, human can explain and can change all the body parts except the brain. So we still there is a lot of work on the brain. There is also a lot of work in the brain-computer interface, so such as passive or active brain interface. So either reading the brain and monitoring the brain signals, such as for telemedicine, for example, or also active brain interface where the user can control either a computer or device just by his brain waves. And also there is a lot of application to the diagnosis of the neurodegenerative diseases, such as the Parkinson, Alzheimer, and of course, the epilepsy in patients. So this is some of the application. So before going further, so I would like just introduce some history about the brain signals recording. So the first brain signals which is recorded was in 1875 by Richard Catton, who is a British physician and physiologist, and he was able to record electrical activity from the brain. So it was from an animal at that time, from different animals, and it was invasive. So he put the electrode directly on the scalp. And what he noticed, so he noticed that there is some activity depending on the action and depending on the mental activity of the animal. So I would say he's showing some picture and he's changing the environment and he observes some changes. So some years later, there is Hans Berger, who is a psychiatry, and he's a professor at University of Vienna. So he was able to measure the first electroencephalography on human scalp. So he put just electrode, this is his device and his laboratory. So he put just some electrodes. So he starts with 2 electrodes on the scalp, and he was able to measure this signal. So here there is just a reference signal, which is a constant signal with a constant frequency and amplitude. And here he was able to observe difference between this part and this part. And he's just asking the subject to close his eyes and open his eyes. And just by this, he was able to record the first wave, and it's called the alpha waves, alpha because it was the first wave. And this is the alpha wave, which appears when the person closes his eyes, and it appears in the occipital sides. I will show you just in a few seconds. And this one was the first EEG recording. So discussing about the waves, so just to continue the idea. So after Hans Berger recording, there is also a lot of research and still ongoing research. So there is many waves of the brain in very low frequency range, so below 100 Hertz. So there is the, let me just move this zoom things away. Yeah. So there is the alpha wave, which is related to relaxation, and it's appearing in the occipital area. So I put this picture here just to show you the different area of the brain. So just the general, because we can divide it in many lobes. So there is the occipital lobe here, parietal lobe, frontal lobe, and the temporal lobe. So each area is responsible for a specific task, as well as we can observe most of the signal in some area. For example, alpha appears in this area. For example, beta is it's appearing in the parietal area here, and it's the sensory and the sensory cortex. And for example, it's when we are busy and we are solving problem. As me now, I'm presenting this slide, so I'm focusing to give you a lot of information. So I think I have a lot of beta waves at this time. There is also other waves like the delta waves, which is appear during sleeping, when we sleep, and also on children. There is also another one which is not cited here, which is the gamma band, which is higher frequency, and it appears when the person is doing some meditation, for example. And of course, for epileptic person, this device can record the spikes of the epilepsy. And from these spikes, there is a lot of application that allow the localization, for example, of the seizure onset or the area in the brain which is responsible to this high activity, which later can be removed. I think I will speak quickly about this later. So I put here this homunculus map, which is kind of artistic representation of the motor cortex and the sensory cortex, and which shows the different area on the cortex, just this part to be sure, not all of the brain. And it shows the different parts of the cortex and its function in the body. So either motor, primary motor cortex or the primary somatosensory cortex. So again, some history. So unfortunately, Hans Berger, at the end of his life, he suicided in his lab. So this is bad news, and because he was nominated many times to the Nobel Prize. But for the EEG, David Cohen, so for the MEG, sorry, there is one in 1916. David Cohen was able to record the first magnetic field from the heart. So he was using the SQUID, the supraconductor sensors, which are developed by James Zimmerman in the picture here. And David Cohen right now is still working in Harvard, Harvard Medical School. And they were able to record the magnetic fields coming from the brain inside this shielded room, because, you know, I will show you just in a few seconds. So the magnetic field in the brain is very, very small compared to the magnetic field surrounding the area. So here, just the first shielded room where the image is recorded, just with one sensor, which is localized here. And today, there is still a lot of working, but now we can have EEG up to 256 channels, as in this picture. I think this one was low. And also the MEG with different sensors like gradiometers and magnetometers. And that allows a high resolution recording of the brain signals, therefore high reconstruction of the source and also better resolution, better representation of the spatial signals. There is also other techniques that I want to introduce quickly, because I will go to speak at least for the MRI. So there is the functional MRI. So F, because it measures the function of the blood or the diffusion of the blood in the brain. And there is also anatomical MRI that allows us to display the different tissues of the brain. There is also the PET scan, which injects radioactive sugar in the blood, and then it will check in which there is high consumption of the sugar, which means there is activity in this area. There is also the NIRS, which is the near infrared spectroscopy, and from where there is injection of the light inside the brain, and it depends on the consumption of the light. We can see how the brain is activated in specific region during a specific task. And of course, the previous methods are non-invasive. There is also the invasive recording, which is where the electrodes are implanted inside the brain. And there is either the stereo EEG or the electrocorticography, where the electrodes are either on the cortex or they are deep in the deep structure of the brain. So this one are invasive recording. So today I will just focus on these two modalities. And if we compare these two modalities compared to the other ones, why? Because there is some reason. So if we speak about spatial resolution, and we means here localization of the source, so we can see that the MRI and the PET scan has the highest spatial resolution in order of millimeters. It's the same thing for the MEG, but the EEG is lowest for the spatial resolution. If we see, for example, the temporal resolution, these two devices are not real time. So we need it takes sometimes in order to localize where is the activity, because it needs to the blood to come to the region in the brain, and then there is this signals that is measured by this device. But for EEG and MEG, they are real time. So at the time of the action, we can record the real time signals. And also there's other features like how easy to use this device, and also the price of the device. For example, EEG, each labs can have it. MEG is about 1 million, and these two devices are higher, more expensive. So just some comparison here. And yeah, regarding to the units of the recording and why we use this shielded room for MEG. So here is just the scaling of the different magnetic field in the current life. So it goes from 10⁻³ Tesla to 32 Tesla, so or more than I never used this one, so 32 Tesla. And we are, for MEG, we are measuring this in this range, so in the 32 Tesla, where with the squids can catch the magnetic field. So compared to the MRI, for example, in the MRI, the gradient or the constant magnetic field inside the MRI, it's around 5, from 0.5 to 7 Tesla, which is very, very high compared to the signal that we measure with EEG. The reason why we need this shielded room. And just regarding the EEG, so with the EEG, we measure the electrical potential, and the electrical potential for EEG is in the range of microvolts. So we don't really need to be in shielded room. So, and since it's a difference of the potential, so we have always the reference in the same area. So if there is noise, it will be propagated all over the electrodes. Same thing as before, but what I want to highlight in these parts, if we compare EEG and EEG in deep, so it's the spatial localization, which is 1 centimeter for EEG and 1 millimeter for MEG. And also what's the difference of the measures. So EEG can catch or can record the extracellular currents or the volume currents. I will discuss about this in a few minutes. Whereas the MEG is able to record the primary current, which is inside the cell. And also, of course, both of them have good temporal resolution. So I will try also to introduce quickly what we are recording, from where it comes. So before, I want just to show you also this brain anatomy, but it's I divided just in 2 tissues. So the brain is composed mainly by 2 matters. So outside of the brain, it's the gray matter, and inside is the white matter. So the gray matter it's about 40% of the brain, and the white matter is about 60%. So if we zoom here, what we can see, so most of the neurons or the cell of the neurons are located on the cortex or on the gray matter. And in the white matter, there is all this extension that we call axons or the fibers, which are responsible for the transmission of the signal between one neuron to the another or one neuron to the body. So all these actions are connecting all the neurons, and also they are going to the spinal cord to control all the rest of the body. And also the white matter is kind of, is here to maintain the neuron and to feed them and to maintain them. Yeah, so what I want to show is just this area where we have the neuron or the source of this brain. So what is going on? So it's similar to what Francesca presented before. So when there is a neuron is activated, so this is a neuron, 2 neurons, and this is the cell, this is the axon, and between the 2 neurons there is space. So when there is a potential action which is generated on the axon that comes from stimulation, outside or inside stimulation. this potential action propagates, will moves on the action, and it lasts just 1 millisecond, and it has high amplitude. When it arrives in this postsynaptic or in the synapse, there is it releases some neurotransmitter. So there is kind of ion that's conduct the signal to the next neuron, and it will either activate or inhibit the next neuron. So it depends on the signal. And then if it's activated, it will generate a new action potential that will propagate to the next neuron and so on up to the organ or the member of the body. So what we measure with EEG is not the potential of action, but because it's very fast, 1 millisecond, we cannot see it. But what we measure is this synaptic potential or the current flow on the potential. So we cannot measure one neuron, but there is kind of spatial and temporal synchronization. So there is not activation in one neuron, but there is a volume of the brain that contain some neurons, so maybe 1 million of neurons, which are activated at the same time. And in this case, we can observe or we can record either EEG on the surface or the magnetic field outside or around the head. So the EEG and MEG is able to observe the postsynaptic potential generated during a specific task. I think I tried to summarize as much as possible the principle, and this is what's happening at the macroscopic and micro and macroscopic level. So now, how to model all of this. So as I said before, I will focus just on EEG and MEG. So the first question, why we are modeling this, and I think part of this question is already answered by the previous speakers. So we want to understand the behavior of the brain or any other phenomenon. We want also to test the different hypotheses and the theory. And of course, we want to understand the different function of the brain, such as the reconstruction of the brain source from the recorded signals. As I said before, for example, if we have some people who are pharmacoresistant to epilepsy, we need to record their signals and then to try to localize the the epilepto zone inside their brain. And also, in order to remove it, and also it's a good way to build the brain connectivity to check and to define which region is connected to which region. And also, it's, of course, it helps to the diagnosis of the different brain diseases. There is another part. Modeling also can optimize the different experiments like TES, TMS, which is already also explained by Francesca. So TMS is just a pulse of magnetic field that we give to the brain in a specific region in order to inhibit or to excite one region. And this region should be defined before. So the position of the coil and its orientation should be modeled before, and we need also modeling for that. Here, this is a TMS, and this is also the TDCS, where we put electrodes, and then we stimulate from one side to another side. And of course, it depends on the application that we are looking for. And here, I'm just showing a result of simulation where we stimulate from one electrode, put on the scalp, and we can see how the current flow inside the head on the different tissues. And we can see, for example, that there is high current on the CSF since it's very conductive compared to the skull, which is very low. So the color here is the amplitude of the current flow. Also, what I want to say here, we use the reciprocity principle, from modeling view, TMS is like MEG, where the source is not inside the brain, but outside of the brain. So it's kind of a reciprocity. So if we have the source inside the brain, we can measure what is going outside. And now if we put the source outside, we can see what is going inside. And the same principle also for EEG. So if we check the source inside for EEG, we measure what is going outside, and here we stimulate from outside and we see what is going inside. So this is based on the Helmholtz reciprocity principle, and from modeling part is almost the same process. And of course, modeling is safe, just what I want to say here. Yeah, there is also this slide. We are also working on modeling intracranial recording. So as I said before, some people with epilepsy, they need to be localized very accurately. Therefore, we need to implant or to put electrode in the brain. And here, this is well segmented brain or with different region. It is more accurate than the previous one. So here, each gyrus is responsible for one task, and this is defined by a lot of fMRI experiments and combining fMRI, EEG, and MEG. And this is just the placements of the different electrodes. And then also we can simulate this kind of scenario in order to help the physician or the neurosurgeon to find the right path, the right direction of the electrodes, where to be put in order to record the epilepsy signals, and in order to localize where is it exactly. So speaking about modeling, in EEG, MEG, we need to speak about the forward problem and of course the inverse problem. So what is the forward and inverse problem? So the inverse problem is the estimation of the brain source. So from the recorded signals, either EEG, MEG, and also stereo EEG, we will try to localize the source within the brain. This is the process of the inverse problem. It's kind of, we have the signals and we are looking to find the antenna that generates the signals. In the other side, the forward problem is the estimation of the signals from a distribution of source in the brain and also a specific position of the sensor or the channel, in this case for EEG. So we need to model. So first we need to solve this forward problem. So we need to define the source model. So how we model the source inside the brain. And also we need to define the medium or the head model, or what we call the volume conductor. So we are stimulating, or we are, activating a source not in the air, but inside the volume conductor. So there is other theory that we will apply for this such problem. Yeah, I add this picture quickly. So solving the forward problem will give us this lead field matrix, or the forward operator that we call we call it the lead field matrix, that link the source to the measure. So here we have all the measures. So V1 is the recording signal in channel 1, V2 in channel 2, and so on. And here S1, S2, S3 is the localization of the dipole or the source in the brain. And this localization has one position and also an orientation, I will say, because it's dipole. And from that, we will need to compute this matrix that map the source to the measure. And this is required to solve the forward problem. And this is also needed to solve and this is required also later to solve the inverse problem. So how to formulate this forward problem? So up to now, we need to model the source. And as I explained before, since there is this current or postsynaptic potential, or the current between the two neurons, so there is displacement of charge, then we can model it as a dipole and a current dipole. And the current dipole is defined by its moment, as explained also before. And this dipole is the primary current, and it's localized only on the position of the source. So for example, here at this position, or inside here, inside the brain. And as I explained before, it should be on the cortex, because the cortex, it's the region that contains the neuron. And since we are inside a conductive volume, so there is also this secondary current, or the volume current, which is due to the conductivity of the medium. So the propagation of the signal will follow the value of the conductivity following this Ohm law, which let's say that the current will be proportional to the conductivity and the electrical field within the brain. And some math, so the total current is the summation of these two currents. And in this field, we assume that we are in quasi-static approximation, which means that there is no loss of the current. So the divergence of the current is equal to 0. So from that will lead to the Poisson equation that's need to be solved on the omega, which is the head model. And also we impose some boundary condition to that model. Therefore, we need to compute the electrical potential, or phi here. So this solution depends, of course, on the geometry of the head, depends on the conductivity, and also the method that we use to solve this problem. And yeah, we can see it clearly in the equations, which depend on the conductivity. So it can be isotropic or anisotropy. And also the source model. We see, I will not focus on that today because it's another topic, but it depends on all these parameters that I try to highlight here. So first of all, as I said, geometry, conductivity, and the resolution method. So first, we need to approximate the shape of the head. So the most simple and the one that is used since years is the spherical head model in 3D. Everything in 3D in this presentation. So we approximate the head by a sphere, and then we project all the sensor on this sphere, and then we put a dipole inside this sphere, and then we use analytical solution. So there is also is closed form solution that can compute the distribution of the potential in spherical medium. This is the basic one. Recently, and let's say the last 10 years, there is the realistic shape, and there is two models. So there is either the surface model, from where we extract only the surface of the model, and we assume that there is isotropic conductivity inside between each two layers. And then we put the dipoles on the cortex, and then we compute this forward operator. And there is also the most advanced one, which is the finite element method. And it's also introduced before this method. So where the head is generated, I will explain it later, that there is tetrahedral, so there is volume between the different tissues. And from that, we can assign more advanced parameters, such as the anisotropy of the different tissues. Also, it depends on the tissues. So as I said, we can assume just there is one tissues, as well as we can use multi-layer sphere, so multiple spheres. So that represent the brain, the skull, and the scalp. Yeah, and of course, as I said before, the conductivity, it can be either isotropic or anisotropic. So before the spherical head model, we cannot assume this anisotropy, but with the recent method that we have, we can include the anisotropy model. So here we have just a coronal view of the brain, and this is just a representation of the tensor. So in 3D, so tensor is the conductivity of the tissues. So the CSF or the liquid cephalorachidien, I forgot. Spinals, yeah, sorry for that. Yeah, so it's mainly water.
Cephalorachidian fluid, probably.
Yes, yes, cephalo, yeah, exactly. Yeah, it's water, and it's mainly the diffusion of the water is mainly isotropic, and it is very high. So the conductivity is very high inside these cavities in the brain, and as well as in between the brain and the skull. There is also this gray matter that I explained before, where the neuron are, I didn't explain it before, but there is different kind of neuron, but we assume also that the gray matter is mostly isotropic, but it's very low compared to the liquid, to the spinals CSF liquids. And what I want to highlight here is the white matter here. So the white matter is not to have the fibers or the, as I showed before, there is this axon that comes from the neuron to the spinal cord here. So there is a lot of fiber that connect the different neuron. So in that case, the conductivity is not isotropic. So the shape of the tensor is not spherical, but it is kind of ellipsoidal. And I will explain also how we were able to compute this anisotropic conductivity for the white matter. And of course, just a repetition, so we have the analytical formulation and we have the numerical formulation. And that's, as I said before, there is a lot of method, but in this field, mainly there is the BEM and the FEM, which is this model. And in my work here, so I will mainly focus to the finite element method modeling for the head. Because as I said, it's the ones that allows integration of more advanced feature, as well as since we have also new method to speed the computation, we are able to implement all these methods in the new software that I'm going to show as well. Yeah, so just a quick overview about the complexity of the model. So as I said before, we can use simple model with 3 layers. So only the brain, skull, and scalp. And we can also add more complexity to the model. And adding more complexity, we add more tissues. So for example, here we have skin, skull, and brain. Here we add the CSF in the blue here, light blue here. And then in that one, we separate the gray and the white matter. So we can see there is now 5 tissues. And also we can go further where the skull or the bone is divided in two layers. So it's most of the time we model the skull as just 1 layers, but it's hard to segment. I will explain it maybe later. So the bone in reality, it has this the compacta, which is in blue, and there is also this spongiform, which is in red here. And it's important because it's affect the EEG signal since it has different conductivity value. And also for the TES, it's important to model this spongia, the spongiosal tissues in the brain. Yeah, so most of the time and up to now, we use the standard conductivity measured in either on animals or from human tissues, but in vitro. So, and it shows in a recent study that it can be different if we measure in vivo and in vitro. So this is just a table of the well-known conductivity of the tissues. So here, this is the list of the compartments. And this is an example of the conductivity that we assign for this compartment. And regarding the white matter, as I said before, this is a fiber tracking method, which is developed also in our lab but by another team. So they were able from the diffusion MRI to extract the fibers and plot to plot them in different colors. So the colors, it means the orientation of the fiber. So green is from left to right, red is from up to anterior superior, and there is the blue as well, but we cannot see it. Yeah, we can show it in 3D. Yeah, so how we do this to convert this diffusion MRI to anisotropic conductivity. So first we need to measure the diffusion MRI, and then we need to extract the eigenvalue, so the eigenvalue and the eigenvector. And then we need just to apply, so this is I simplified, so we need to compute scaling factor. There is this paper, and it's up to now, we are working also on another topic, but how to measure the conductivity using invasive recording. But there is just a linear scaling factor between the diffusion tensor to the conductivity tensor sigma here. Yeah, so I think I just highlighted the main different step. I didn't put, if we have time, I have the more in the annex, but I try to stay in the time. So now what I want to show you, so as the title says, so modeling tools. So mainly I will discuss Brainstorm and its plugin and all this pipeline that I presented, which is now implemented since now 2 months, and it's available for the people in this field. So first, what is Brainstorm? So Brainstorm is one of the software which are open source and free for the user, for the neuroscience community to model EEG, MEG, and all the neurophysiological data. So it's based on MATLAB and Java. It's multi-platform. So you can use it also if you don't have MATLAB. So MATLAB is a problem for a lot of people right now, but Francois, or the main person in charge, he was able now to use it without any MATLAB license. So it's very easy to use. So you don't need any programming skills. So you just click drag and drop, and you have just to follow the tutorials. So there is a lot of online documentation, and we are very active on workshops. So there is a lot of workshops that are organized. For example, there is a workshop after tomorrow, Friday. And there is also forum and GitHub, and where we answer all the questions and solve the problems. And of course, every day you can update the software because we are still adding a lot of feature. So this is just a quick overview. So this is visualization of ECOG channels on the cortex. This is SEEG implantation. All this is obtained from real data. So we need to have the MRI of the subject and the CT that's map or that's localize all these points or the contacts. And then we can also, of course, EEG, MEG, and also we can monitor, for example, with camera video, we can observe here there is a rat inside the box that is moving, and we record its signals. And some people use it to monitor some epileptic person, so they can record 24-hour video, and they have their signals, and then they can show if the signals map to seizure, for example. And also there is a support FNIRS. So the workshop of Friday is about FNIRS. I can share with you a link if you are interested. So this is the graphical interface. So everything is done from the graphical interface, and you don't need any programming skills. So we can display the signals here, the signals with butterfly representation. Here the topography on the sensor, here the channels, and we can click on the channels and highlight it here. We can compute the inverse problem and display the source. I will show you quickly. And of course, if you are experts in programming, there is also scripting environments where you can convert any things that you do on the graphical interface to a script. And then you can process big data, you can send it to server, you can do further computing than just from the interface. So the main workflow, as I said, so we need the individual anatomy of the subject. We need the recording and the real localization of the sensor. I didn't focus a lot on the sensor, but when you have the MEG, it's automatically loaded because the location of the MEG are in the machine. But when you have EEG, so EEGs depend on the shape of the head. So we need to digitize the localization or the 3D position of each sensor. Then we need to do some co-registration, and we check that the sensor fits the shape of the head. And from that, we will do this, what I explained, the forward and the inverse problem, and then we can do the source estimation from the signals. And of course, there is a lot of feature that can be done from, for example, we can average the signals, do a lot of statistics analysis, frequency domain connectivity, and a lot of features that's still ongoing. So today I will just focus on these parts, which is mainly the forward problem as I explained before. So, and it will be based on the finite element methods that I showed before as well. So mainly there is two phases. So the phase one is the FEM head model generation. So we need individual, I didn't explain it as well. So in order to localize accurately the source inside the brain for an individual, we need to use his own MRI. So we need to to acquire this MRI, and then we built his own model with its own data in order to be more accurate in the modeling. However, if in some cases, and most of the cases, the people, they don't have their MRI, so there is a lot of templates which are validated by the community that can be used, and they are freely available in the software. So the phase one is the head model and conductivity tensor generation. I will explain it. And the second phase is the computation of the forward problem, or the computation of the lead field matrix that I showed to you before, and all this using the finite element methods. I think I will explain this phase one step by step. So the first one, we need to import the MRI of the subject. I don't know if you are familiar with the MRI, but the MRI has a lot of sequence, and the well-known sequence is the T1 and T2 weighted MRI. So the T1 allows us to see the difference between the white and the gray matter. And however, it's difficult to distinguish the skull and the CSF. So therefore, it's better to use the T2, but combining the two MRI, we can segment the MRI. So, so both of them. So yeah. So what I want to say, so the first step is the segmentation. So what is the segmentation? It's kind of statistical affiliation. So each voxel, so the voxel is, the MRI is kind of cube, and each cube has an intensity, and this intensity represents one tissues. And in reality, there is a lot, not 100, but let's say 20 or more tissues in the head. But the segmentation, what we need is just mainly the 6 or the 5 tissues. So the skull, white, gray, and the CSF. So for that, there is some tools that are already integrated in Brainstorm that allow the segmentation. So we integrate this, so the user just use Brainstorm and everything is documented. So the first step is the segmentation of the T1 and T2. This is the list of the tool or the plug-in which are already integrated. From the segmentation, we can generate the FEM mesh. And as I said before, the mesh can be either tetrahedral or hexahedral. I think here I did zoom. The tetrahedral is kind of a pyramid with 3 faces or 4 faces. And here, this is the cube or the hexahedral mesh. So both models are available. So, and we have also some advanced computations such as node shifting and other options. Then, in order to compute the anisotropy or the conductivity tensor, we need the diffusion MRI, which is also another sequence of the MRI where we need the gradient and we need to acquire the tensor. We need, it's another sequence which is recorded with different gradient orientation, and it depends on the energy located in the voxel. It can give us how the waters move in the white matter and also in the others, but we need just to focus on the white matter since, as we said before, the white matter is the anisotropic tissues in the brain. And everything from interface. So we need just to specify the conductivities of the tissues as I showed before on the table. And then we say which tissues that we need as isotropic here. And then we specify the method of conversion from the interface. And from that, we can compute the tensor. So here we have the diffusion tensor. This one is the conductivity tensor. And as we can see that the colors are the same, and the colors show the orientation of the ellipsoid or the orientation of the main eigenvector with its main eigenfactor. The second phase, so once we have the head model and the tensor, so we have the full head model. Second phase is the computation of the FEM head model or the forward problem. So we need to specify the localization of the dipole. So mostly we put the dipole on the cortex, and we have a lot of options that can check that the cortex fits exactly the MRI of the person. And then we need to co-register to the localization of the sensor EEG or MEG. And from that, we can just specify some option for the FEM computation. So we have the last one. So we use this continuous Galerkin, this continuous Galerkin, and all this is implemented. It's part from my PhD thesis and also from the German group, which developed all this tool. And I did just small contribution to this work, but my contribution was for Brainstorm. And then we can compute the lead field, and we have also option to visualize the lead field. So the lead field are, in other word, is the sensibility of the sensor to a specific source in the brain. So here we can see the orientation of the vector are coming from the reference to the target. So it's it's similar to what I said before. It's kind of TDCS. If, for example, we want to see how the TDCS stimulation pattern inside the brain is, it should be like this. So we inject the current here, and then we can see what is going inside. If we want to target any region, we can see how this region will be sensitive to this two location. Same thing here. This is for EEG. This is for MEG. And we can see this is a nice pattern of current from a magnetometer. And the same thing, if we stimulate with TMS, we can see exactly the same pattern, which is based on the reciprocity, as we said before. So from that, we can apply the standard inverse modeling in Brainstorm. So from the signals, we can try to build the source. As you can see here, there is a dipole here. You can see there is the two pole of the magnetic of the dipole. This is EEG. This is, sorry, from 2 Tesla. This is MEG. This is EEG, and both are perpendicular. And we can see that it was able to localize either the current density or the dipole fit. We can see there is a dipole here. And if we fit it here, we can see it can have the same pattern. Yeah, I think I'm done. I think maybe, I'm not sure if I'm in the time, but this is the last slide. So I tried to give you an overview about the brain and electromagnetic brain, electromagnetic modeling of the brain, kind of different of what is presented before. So this pipeline is implemented to Brainstorm, and we use a realistic head model for better modeling of individual person. We use all these MRI tools and function to model this, the individual lead fields. And everything is available, and we have this website where we can find tutorials, examples, data, and everything. And also we can contact the team. So the team is a big team. So we have a group, I'm here. So at USC, we have a big collaboration with Germany at Münster. We are also at Houston with John Mosher, one of the leaders in MEG. We have also some colleagues in Harvard, and also the Brainstorm teams, which is from us, McGill, MIT, and also Texas. And this is just some picture of all the team. Thank you, and I'm open to any other questions.